occur here. These include mechanical breakdown of food by chewing, initiation of starch digestion by mixing food with secretions from the salivary glands, and propulsion of food to the pharynx by the tongue. The lips and cheeks are important in keeping food in the mouth and between the teeth when chewing. Anchoring the lips to the gums are the superior labial frenulum and inferior labial frenulum. The lingual frenulum attaches between the tongue and the floor of the mouth. The vestibule is the space located between the external cheek and lips and the internal gums and teeth. The teeth are anchored in the gum-lined alveolar processes of the maxillary and mandibular bones. During development, we normally have two different sets of teeth. These two sets can be seen on this child's skull. The first set of teeth to erupt is the deciduous set, which consists of 20 teeth. It is replaced by a full set of 32 permanent teeth by the time we're in our mid-20s. The types of teeth we have as an adult can be seen on this adult skull. These include the central and lateral incisors, cuspid or canine, first and second premolars or bicuspids, and the first, second, and third molars. When the head is sectioned in this mid-sagittal view, it's easy to see how large the tongue is and how much space it takes up in the oral cavity. The tongue is formed from skeletal muscle and, like the oral cavity, is lined by non-keratinized stratified squamous epithelium. The bony hard palate and muscular soft palate separate the nasal and oral cavities. The hard palate consists of bony projections of the maxillary and palatine bones. The soft palate is a muscular structure whose most posterior portion is the uvula. It attaches to the wall of the oropharynx by the palatopharyngeal arch. The palatoglossal arch, which lies more anteriorly, attaches the soft palate to the tongue. The palatine tonsil resides in the space between the two arches. The lingual tonsils are located near the root of the tongue. Three pairs of salivary glands produce saliva which cleanse the mouth, moisten food in preparation for swallowing, and dissolve small food substances for tasting. Saliva also contains amylase, which initiates chemical digestion of starch. The submandibular gland lies below the mandible. The opening of the duct from this gland is found in the mouth near the lingual frenulum. The sublingual gland is smaller and deeper in position and transmits its secretions through several ducts which open into the floor of the mouth. The parotid gland overlies the masseter muscle anterior to the ear. Its duct penetrates the buccinator muscle and empties into the mouth near the second upper molar tooth. The pharynx, a skeletal muscular tube lined by epithelium, transports air and food and is divided into three regions by several anatomical landmarks. The nasopharynx, lined by pseudostratified ciliated columnar epithelium, lies posterior to the nasal cavity and extends to the level of the soft palate. The oropharynx is the portion of the pharynx which lies behind the oral cavity between the soft palate and the hyoid bone. The laryngopharynx extends from the level of the hyoid bone and divides into the larynx anteriorly and the esophagus posteriorly. The protective non-keratinized stratified squamous epithelium which is found in the mouth continues to line the oropharynx and the laryngopharynx. As previously described, the gastrointestinal tract is a long tube which is continuous from the mouth to the anal canal. Each region of the GI tract varies in both its structure and function during the digestion of food. The four basic layers of the tract include the mucosa, submucosa, muscularis, and serosa. The innermost layer which surrounds the lumen is the mucosa. It protects the body from foreign invaders, secretes mucus which lubricates the tube, and absorbs digestive products into the blood. The mucosa is composed of three different layers. The moist epithelial lining of the lumen is usually formed from simple columnar epithelium with goblet cells and mucosal glands. Highly vascularized, loose areolar connective tissue forms the middle layer of the mucosa called the lamina propria. The capillaries in this layer receive absorbed nutrients from the tract. The lamina propria may also contain a few protective lymphatic nodules dispersed throughout the tissue. The outermost layer of the mucosa is a thin layer of smooth muscle called the muscularis mucosae. The submucosa, a layer of dense connective tissue, lies external to the mucosal layer of the GI tract. It is composed of blood vessels, the submucosal plexus, which is a part of the enteric nervous system, lymphatic vessels and nodules, and elastic fibers. It is also the site of submucosal glands, which secrete substances that are transported through the mucosal layer and deposited into the lumen during digestion. The muscularis layer, sometimes referred to as the muscularis externa, typically consists of an inner circular layer of smooth muscle and an outer longitudinal layer of smooth muscle. The myenteric plexus, another part of the enteric nervous system, is located between these two muscle layers and controls their contraction. Activity of the muscularis layer gives rise to the various motility patterns found in the GI tract. The portions of the gastrointestinal tract which are suspended intraperitoneally within the abdominopelvic cavity are covered by a layer of serosa. It is formed from connective tissue and simple squamous epithelium called mesothelium. This serosal membrane is also called the visceral peritoneum when it covers the organs within the cavity. The esophagus, a muscular tube, conducts food from the laryngopharynx to the stomach. In the upper thorax, the esophagus, about 10 inches in length, lies posterior to the trachea. It descends in the thorax, travels behind the heart, and enters the abdomen by passing through the esophageal hiatus in the diaphragm. The histology of the esophagus has many similarities to the general histology of the gastrointestinal tract, but it also differs in several ways. Instead of the simple columnar epithelial lining found throughout most of the tract, the esophagus is lined by a continuation of the protective non-keratinized stratified squamous epithelium which covers the laryngopharynx. The muscularis of the esophagus is skeletal muscle in its upper third and smooth muscle in its lower third, with a mix of skeletal and smooth muscle in the middle third. In addition, an adventitia of fibrous connective tissue helps to anchor the tube to the thoracic wall instead of a serosa. The stomach is the expanded portion of the GI tract which primarily serves as a storage organ. In addition, protein digestion begins here, and there's some absorption of alcohol and drugs. Now, let's locate the stomach on our specimen and review its relationships to other organs in the abdomen. The abdominal wall has been opened to allow us to view the contents of the abdominopelvic cavity. The stomach lies mostly to the left of the midline and directly below the diaphragm. Lying to the right and covering over part of the stomach is the liver. When the liver and diaphragm are reflected away from the stomach, the opening of the esophagus into the cardia is seen. Lying to the left of the cardia is the fundus, which is the most superior part of the stomach. The large middle portion is the body. The stomach begins to narrow at the pylorus region to empty into the duodenum through the pyloric sphincter. The stomach has a characteristic C shape. The concave medial surface is the lesser curvature, and the convex lateral surface is the greater curvature. The lesser omentum, a peritoneal reflection, attaches between the liver and the lesser curvature of the stomach and the duodenum. The large apron-like structure which hangs down from the greater curvature of the stomach to cover the small intestine is the greater omentum. It contains collections of fat and lymph nodules filled with lymphocytes and macrophages. These cells are part of the immune system which monitors our body for foreign invaders. In this specimen, the stomach is open so we can examine the inside. When the stomach is empty, large longitudinal folds of the mucosa and submucosa called rugae are present. As the stomach fills, these folds flatten out to increase stomach capacity. The wall of the stomach contains the 4 layers characteristic of the GI tract, including the mucosa, submucosa, muscularis, and serosa. The major specializations in the stomach region of the tract are the mucosal epithelial lining and the muscularis layer. The simple columnar epithelium with goblet cells of the mucosa layer secrete a thick coat of alkaline mucus to help protect the lining of the stomach from damage by acidic gastric juice. The epithelial lining is organized into gastric pits which penetrate deep into the lamina propria. These pits lead into the gastric glands where several cell types contribute secretions to the gastric juice. Mucous neck cells produce mucus. Parietal or oxyntic cells produce hydrochloric acid and intrinsic factor, and chief or zymogenic cells produce an inactive form of pepsin, a proteolytic enzyme. Other cells in the mucosa, collectively referred to as enteroendocrine cells, produce a variety of hormones which influence digestive processes throughout the GI tract. The muscularis layer is also modified. There are 3 layers of muscle instead of the 2 layers which are characteristic of most of the remainder of the tube. The innermost layer is obliquely oriented, the middle is circular, and the outer layer is longitudinal. This specialization of muscular arrangement permits better churning and mixing of food with gastric secretions during digestion. The small intestine is where chemical digestion is completed and most of the absorption of nutrients takes place into the blood. It has 3 segments, the duodenum, jejunum, and ileum, which will now be examined in our specimen. To see the small intestine, the greater omentum and part of the large intestine are retracted. The small intestine extends about 21 feet in the cadaver but is shorter in the live individual because of smooth muscle tone. The duodenum, the proximal part of the small intestine, is about 1 foot in length. It begins at the pyloric sphincter and is the site where secretions from the pancreas and bile from the liver and gallbladder enter into the GI tract. Most of the duodenum lies behind the peritoneum and is not visible in this specimen. The second segment of the small intestine, the jejunum, is the continuation of the duodenum and is about 8 feet long. It is a highly coiled tube that transports digestive material into the ileum. The ileum, about 12 feet in length, is the longest portion of the small intestine. It terminates at the ileocecal valve, which separates the small intestine from the cecum of the large intestine. The mesentery, another peritoneal reflection, suspends the jejunum and the ileum in the abdominal cavity from the posterior body wall. Blood vessels, lymphatics, and nerves travel within the mesentery to reach the small intestine. So that we can see the architecture of the blood vessels supplying the tract, the mesentery has been opened in this specimen. Branches of the superior mesenteric artery supply blood to the jejunum and the ileum. Arched vessels, called arcades, are formed from adjacent branches of these vessels. Straight vessels, called vasa recta, arise from these arcades. The arrangement of arcades and vasa recta are different in the two parts of the small intestine. The arcades are fewer in number in the jejunum than in the ileum, and the vasa recta are longer in the jejunum. The walls of the small intestine have the same 4 layers as the other parts of the GI tract. However, there are some important modifications of the walls that facilitate absorption of nutrients by increasing the surface area 600-fold. One of these modifications is seen in our specimen when the small intestine is opened. These deep, permanent circular folds are called the plicae circulares. This pattern results from folding of the mucosa and submucosa layers in the intestinal wall. Finger-like projections of the mucosa called villi further increase the surface area for absorption. The villi are lined by simple columnar epithelium with absorptive cells, goblet cells, and enteroendocrine cells. The absorptive cells have tiny projections of their surface membranes called microvilli, which make up the brush border and also increase surface area. Brush border enzymes complete the digestive process of disaccharides and small peptides. Each villus has a core of lamina propria which contains a blood capillary network and a lymphatic capillary called a lacteal. Protein and carbohydrate breakdown products are absorbed into the capillary bed, and fatty substances are absorbed into the lacteal. Located between adjacent villi are intestinal crypts whose lining cells secrete intestinal juices. Similar to most of the GI tract, the muscularis layer in the small intestine contains an intercircular and outer longitudinal layer of smooth muscle. The serosa, or visceral peritoneum, covers the majority of the small intestine, except for a portion of the duodenum, which is retroperitoneal. The liver, gallbladder, and pancreas are accessory organs which are not part of the gastrointestinal tract, but produce substances that empty into the duodenum through a common duct to aid digestion. The liver is the largest gland in the body. The bulk of the liver lies to the right of the midline below the diaphragm, protected by the rib cage. The liver is divided into 4 lobes, 2 of which can be seen here. The largest, the right lobe, is separated from the left lobe by the falciform ligament. This supporting ligament attaches to the diaphragm and the anterior body wall. Lying in the inferior border of the falciform ligament is the round ligament, or ligamentum teres. This fibrous cord represents the remnant of the umbilical vein in the fetus. Inferior to the liver lies the gallbladder, a storage sac for bile. In order to see parts of the liver more clearly, we have removed it from our specimen. From this anterosuperior view, we can identify the right lobe, left lobe, falciform ligament, ligamentum teres, and gallbladder. The other 2 lobes of the liver can be seen best when the liver is observed in a postero-inferior view. We will begin by identifying the right lobe and the left lobe again. The caudate lobe lies near the inferior vena cava. The quadrate lobe is located near the gallbladder. We can also see the ligamentum teres in this view. A liver lobule, the functional unit of the liver, is hexagonal in shape, with each of the 6 corners of a lobule containing structures called triads. Each triad is composed of a branch of the hepatic artery which carries oxygenated arterial blood to the liver, a branch of the hepatic portal vein which carries venous blood draining from the digestive tract, and a bile duct. The liver cells, or hepatocytes, are arranged in rows which radiate from a central vein and are separated by small spaces called sinusoids. Arterial blood and venous blood flow from the periphery of the lobule toward the central vein through these sinusoids. Hepatocytes lining the sinusoids process the nutrients in the blood. Hepatic macrophages, or Kupffer cells, remove old blood cells and foreign material from the blood. In addition to processing blood nutrients, hepatocytes also produce bile, which is collected by small canals called canaliculi. Bile is transported through the canaliculi away from the center of the lobule toward the bile ducts located in the triads. Bile exits the liver through the left and right hepatic ducts. They unite to form a common hepatic duct, which joins with the cystic duct to form the common bile duct. If bile is not needed for digestion, it is diverted to the gallbladder, where it is stored and concentrated by removing water and ions. When bile is needed for emulsifying fats in the digestive tract, it leaves through the cystic duct and empties into the common bile duct. This duct joins with the main pancreatic duct to form the hepatopancreatic ampulla. It opens into the duodenum at the major duodenal papilla. The hepatopancreatic sphincter controls the movement of bile and pancreatic secretions into the duodenum. 2 ducts collect pancreatic secretions and empty into the duodenum. The course of one of them, the main pancreatic duct, was described with the common bile duct. The other, the accessory pancreatic duct, is smaller and opens into the duodenum more proximally than the larger duct. Pancreatic secretions contain a variety of enzymes that digest starch, fats, and proteins. Both bile and pancreatic secretions are alkaline-rich to neutralize stomach acids emptied into the duodenum. To examine the location of the pancreas on our specimen, we will reflect the diaphragm and liver. The pancreas lies posterior to the stomach. In order to see it more clearly, the peritoneal covering of the pancreas has been removed. The head portion of the pancreas is partially surrounded by the curved duodenum. The body is the central portion, and the tail is the tapered portion. Notice the close relationship of the tail of the pancreas to the spleen. The large intestine is the terminal portion of the GI tract and functions to absorb water and eliminate indigestible foodstuffs. The large intestine begins at the cecum and extends to the anal canal. The large intestine, about 5 feet long, is wider in diameter than the small intestine. The large intestine begins at the cecum. Extending from this pouch-like portion of the large intestine is the vermiform appendix, actually part of the immune system. From the cecum, the ascending colon travels along the right side of the abdominal cavity and makes a turn at the right colic flexure near the liver. The large intestine continues as the transverse colon. It makes another turn at the left colic flexure near the spleen and continues down the left side of the abdominal cavity as the descending colon. Upon entering the pelvic cavity, it becomes the sigmoid colon, named for its S shape. The sigmoid colon empties into the rectum. The large intestine terminates at the anal canal, which is open to the exterior at the anus and cannot be seen here. The ascending and descending portions of the large intestine are retroperitoneal in position and lie posterior to the peritoneum in the abdominal cavity. Their peritoneal coverings have been removed in our specimen. The transverse colon is anchored to the posterior body wall by its mesocolon. Similarly, the sigmoid colon is anchored to the posterior